Hey everyone, welcome you all to Bio School. This is Gayatri. So in today's session, we are going to discuss about a very important topic, and the topic name is excretion of Lac Operon in Merozygotes. In my previous video, I have explained operon concept in details. I will drop the link in the description box down below, so please check out that video too. Let's proceed and know in details about merozygotes as well as the expression of Lac Operon in merozygotes. So first, what is merozygotes? Merozygote, it is a bacterial cell that is partially diploid. Okay. So how the merozygote formed? Merozygote, it forms when the F prime cell it conjugate with F minus cell. So here the F prime cell that is able to grow on a lactose medium, whereas the F minus cell that is unable to grow on a lactose medium. So first know what is the F prime cell. In F prime cell, in addition to the bacterial chromosome, there is a presence of the F prime factor. It is a F factor, that is the fertility factor. Along with that, you will find some bacterial chromosome. Whereas in F minus cell, the fertility factor or the F factor, it is absent. So when there is a conjugation, then there will be DNA transfer that lead to the formation of a partial diploid cell. Here you can see when there is a gene transfer, that the bacterial gene, that is the lactose plus, it get transformed into the F minus cell and it lead to the formation of a F prime X conjugate, where there is a presence of both Lac minus as well as Lac plus gene. Okay. Now the bacterial cell, it is a partially diploid. It is diploid for a particular gene, that is the lactose. Now you will see if there is a mutation of Lac genes, then what will be its outcome? So first, Lac I. You know the Lac I gene, it encodes repressor protein. So if there is a mutation of Lac I gene, then either it do not synthesize the repressor or it will synthesize the repressor which is not able to bind to the operator. Okay, its function is to synthesize the repressor, but when the Lac I gene it is mutated, then at that time it cannot synthesize a repressor. If it synthesizes the repressor, then that repressor is not able to bind to the operator. Okay. The second one is Lac IS. Okay, that is the Lac super repressor. Lac super repressor, it results in change in the conformation of repressor so that the repressor it cannot bind to the inducer. Okay, and the inducer for Lac Operon it is allolactose. Lac I D minus, that is the Lac mutation of dominant negative. In this condition, then it will produce non-functional repressor monomers. There is a mutation Lac OC, it is the mutation of operator. If the operator it is mutated, then it does not allow the binding of a repressor molecule with operator. That lead to the continuous gene synthesis, that is expression or constitutive gene expression. Okay. And then last one, it is the Lac P minus, means here the promoter is mutated. If the promoter is mutated, then RNA polymerase it cannot bind to the operator region. So for solving any question related to expression of Lac Operon in merozygote, you need to have a clear idea about what is merozygote and what are the outcomes if the Lac gene it get mutated. Then you can solve any type of question related to this topic. Now we will see some cases. Case 1. Mutation of regulator gene. Lac I, it is get mutated. Okay. So this one is the E. coli chromosome. And in the E. coli chromosome, this one is the Lac Operon. Here there are the presence of 3 structural genes. First one is the Lac Z, Lac Y and Lac A. And here it is the operator region. Operator, then overlapping with the operator, that is the presence of promoter region, and the transregulator gene or the Lac I gene, it is mutated. That's why here it is written as the I minus. If the Lac I gene it is the mutated, Lac I gene it codes for the repressor, but now it is mutated, so it cannot produce the repressor protein. Okay. So it cannot produce the repressor protein, or if it produce the repressor, then it cannot synthesize the repressor, or if it synthesize the repressor or the mutant repressor, then that mutant repressor it cannot bind to the operator region. If the repressor it cannot bind to the operator region, then RNA polymerase it will bind to the promoter region, that lead to the continuous gene expression or constitutive expression. Okay. Now we will see the similar condition, that is the mutation of Lac gene in Merozygotes. So this one is the E. coli chromosome. Okay. So this point is the operator. These are the structural genes, and this is promoter region, and the I gene it is mutated. So it is written as the I minus. This is about the E. coli chromosome. And the other chromosome, it is the Merozygote means partially diploid. So other chromosome. So here the Lac gene it is not mutated. Okay, here the Lac gene it is unmutated, and there is the presence of promoter, operator region, and the structural genes. So E. coli chromosome I gene or Lac gene it is mutated. That means it will produce a mutant repressor protein. But on the other hand, the A prime factor it will produce the normal repressor. So now the repressor it is produced as a monomer, then it forms a tetramer condition. This tetrameric repressor it can bind to both of the operator. It can bind to the operator of E. coli chromosome as well as the operator of A prime factor. So as both the operator region are occupied by the repressor protein, so RNA polymerase cannot bind to the promoter region. As RNA polymerase cannot bind to the promoter region, then that leads to the repression of both the operants. Okay. In the second case, mutation in the operator OC it is mutated. What I told you before, if the operator region it is mutated, then the repressor protein it cannot bind to the operator. Okay. So in E. coli chromosome, repressor it is I plus, this one is the promoter, then operator here it is mutated, that is OC. Though the repressor gene or Lac gene it produce the normal repressor, but this normal repressor it cannot bind to the operator region, as the operator region it is mutated. So if repressor it cannot bind to the operator, then RNA polymerase it will bind to the promoter region, and it will cause the constitutive expression of gene. Okay. Now let's have a look on the Merozygote. In E. coli chromosome, okay, E. coli chromosome the operator region it is mutated, but in the other chromosome operator it is normal. Okay. So the I gene, Lac gene, it will produce the repressor. This repressor it cannot bind to the E. coli chromosome operator as it is mutated. Okay, but it can bind to this normal or unmutated operator region. So when operator region it is bound by the repressor, then what will happen? This portion it will be repressed. Then this gene it will be repressed. On the other hand, the E. coli chromosome it will constitutively expressed. Okay, case number 3, mutation of regulator gene. Regulator gene it is mutated, Lac ID minus. So if the Lac ID gene it is mutated, then it will produce the non-functional repressor monomer. So see, this one is the E. coli chromosome. Okay, so here it is the I minus D, means it will produce the non-functional repressor monomers. But on the other hand, the other chromosome, this one is the I plus. Okay, means the repressor gene it is in normal condition, so it will produce the normal repressor. See over here, in the tetramer condition you will find the 2 are functional repressor monomers and 2 are non-functional repressor monomers. So is the repressor it is non-functional, so it cannot bind to either of the operator. So if the repressor it cannot bind to either of the operator, then both the promoter region it will be bind by the RNA polymerase, and that will cause the expression of gene, which is known as the constitutive expression. Okay. So now we'll solve some previous year CSIR NET question related to this topic. So first question. With reference to the Lac Operon, what will be the phenotype of an E. coli strain having genotype? Okay. So see, is the genotype, it is I minus, okay, then the genotype is I minus O plus Z plus Y minus, and in the F prime factor, the genotype, it is I plus OC, that is the operator region, it is mutated, Z minus and Y plus. So here it is asking about the expression of for both beta galactosidase and lactose permease. Beta galactosidase, it is expressed, it is encoded by which gene? It is encoded by the structural gene Z, right? And lactose permease, it is encoded by the Lac A gene. So this is the genotype, that means it is a merozygotic condition, okay, means partially diploid E. coli cell. So in the first pair or the E. coli chromosome, I gene, it is mutated. If it is mutated, then it will produce the mutant repressor. The mutant repressor, it cannot bind to the operator region. If it is cannot bind to the operator region, then the RNA polymerase, it will bind to promoter region, then that lead to the expression of gene. Okay. Then next comes to this one. Here it is I plus, that means it is the normal Lac I gene, it will produce the repressor, right? And this repressor, what are produced, this repressor, it can bind to this operator, right? So the expression of gene here, so the expression of gene here depends on the presence or absence of the lactose molecule, right? If the lactose molecule, it is present, then what will happen? If lactose is present, then it will cause the transcription of the gene, because lactose, it will convert it into allolactose and it will bind to the repressor. So the repressor, it cannot bind to the operator region, right? Let's consider if the lactose in the medium, it is absent. Okay, if the lactose is absent in the medium, then what will happen? Then this repressor, it will occupy the operator region. As it will occupy the operator region, then RNA polymerase cannot bind to the promoter region. So the Lac Operon, it will be switched off. Okay. But the repressor, it cannot bind to this operator, because this operator, it is mutated. So the repressor protein can never bind to a mutated operator, and that always lead to constitutive gene expression. Here it will lead to constitutive gene expression. It leads to inducible gene expression, right? Because the expression, it depends on the presence or absence of lactose molecule, which is the inducer. So it is called the inducible gene expression. Now we'll see the genes. See, so it is asking about Z plus, so beta galactosidase. So what do you think? Beta galactosidase, it is a inducible, okay, inducible gene expression. And lactose permease. See, here the Y gene, it is absent, minus, that means in the past, that means here it cannot synthesize lactose permease. Okay, look over here. What will be the expression of beta galactosidase? If Z gene is absent, then there will be no expression of Z gene. But see, Y plus means lactose permease, it will be synthesized. And how it will be synthesized? It will be synthesized constitutively. So what is the correct answer? Correct answer is that inducible expression of beta galactosidase and constitutive expression of Lac permease. The correct answer is option C. Okay. The next question. In context to Lac Operon, if bacterial strain P1 and P2 with genotype, okay, so here it is given the genotype of P1 bacterial strain is OCI plus Z minus, and P2 strain is O plus I minus Z plus, and D, it is a merodiploid. Okay, merodiploid, it is produced from these two strains. Then it is asking about the expression of Z gene. Okay, expression of Z gene in all these three strains. Okay. So first point, if you get any questions from Merozygotes, or then first you have to draw the operon. So here in the operon is the OC, that means operator region, it is mutated. Operator region, if it is the mutated, though the repressor gene or I, it will produce the repressor, but this repressor, it cannot bind to the operator region. As it cannot bind to the operator region, then that leads to the constitutive gene expression in P1 strain. So what is this asking? It is asking about the expression of Z gene. But see, here the Z gene is absent. Z minus means there is no Z gene. Is off, or there will be no expression of Z gene in P1 strain. Let's move to the P2 strain. Here you can see the P2 strain I minus. I minus means the repressor gene, it is mutated. If it is the mutated, then it will produce the mutant repressor, and the mutant repressor, it cannot bind to the operator region. Okay, so mutant repressor, it cannot bind to the operator region, then that will lead to the constitutive gene expression. Okay. So here the Z gene expression in P2 strain is constitutive expression. Let's see the option. Okay. So let's eliminate the option C, because here it is showing the P2 is inducible expression. No, it will be constitutive expression. So correct answer either option A or option B. So now we'll see the merozygote condition. So in the merozygote condition, the repressor protein, it will produced here, and this repressor protein, it cannot bind to this mutant operator, but this repressor gene can bind to the operator of P2 strain. So when it will bind to the P2 strain, it will bind to the P2 strain if in the medium lactose or the inducer allolactose is absent, right? So if the lactose is absent in the medium, then only the repressor, it can bind to the operator region, and that will switch off the expression of structural genes. But if in the medium lactose is present, then what will happen? This repressor, it cannot bind to the operator region. The repressor, it will bind to the allolactose, and RNA polymerase, it will bind to the promoter region, that will turn on the gene expression, right? So in merozygotic condition, the expression of the Z gene, it is inducible. Okay, because that expression, it depends on the presence or absence of the inducer. So okay, so let's see the option. We got the D is inducible expression. So the correct answer is option A. Okay. The P1 strain, no expression. P2 strain, constitutive expression. And D, or the merodiploid strain, it has the inducible expression. Thank you so much for watching this video. If you still have any confusion regarding merozygotes and expression of Lac Operon in merozygotes, then please put a comment in the comment section down below. I will try to solve your question. Thank you so much for joining me today. If you like this video, then please share and don't forget to subscribe my channel.